Welcome to everybody, and thank you very much for joining me today on today's webinar on the topic of quantification of target elements in individual cells by single cell ICP-MS for medical applications. And I want to especially focus on the new instrumental capabilities which are existing at that point in time to make a difference for the outstanding research questions. My name is Helmut Ernstberger. I'm a field application specialist in the inorganic product lines for PerkinElmer. I'm especially focused on the export territories on the periphery of Europe and on Spain. The agenda for today's talk is the introduction to single cell ICP-MS. Here we want to cover what the technique can offer. We want to understand the requirements in terms of the hardware and the software in order to make those measurements. And for that, we also need to look into the theory for single cell ICP-MS. And I will also take a step back there, looking into single particle ICP-MS, because it allows us a better appreciation of where the differences in terms of the requirements lie. Then I want to move on to the medical applications. So I have a number of examples there. It's the single cell ICP-MS analysis for platinum. So that would be a reagent which would be accumulating in the cells, a platinum-based reagent, and we would analyze the content of platinum in the single cells to measure its distribution. Secondly, it's an example about nanoparticles accumulation in single cells, and that is in respect to gold. Nanoparticles are a carrier for reactive entities in the world of cancer treatments, and for that reason receive special interest. So the application in this respect is covered with that line item. Next in line is the neutron capture therapy and the special case for boron. Boron in the neutron capture therapy is receives a wide attention, and there's a number of important research applications which need to be currently investigated. And for that, we have new instrumental capabilities which will help in addressing those research questions. So for neutron capture therapy for boron and also for sulfur, we want to discuss what the new instrument lineup can bring in terms of an increased toolbox to address those research questions. So where does single cell ICP-MS differ in the conventional approach to measure the accumulation of a certain target element in cells? That's the question for this slide. Traditionally, we would look at the metal content at the cellular level by digesting the complete cell population and determining the average of the target element in the digest solution. So we would get a single value, which is the average of all cells. In single cell ICP-MS, on the other hand, we are able to measure the discrete quantity of target elements in each entity, so in each individual cell. And therefore, we get not only the average value, but we also get the average value of the population, but we also get the individual values for the cells which contain the target element. And that's pretty much the same situation when we look at nanoparticles. The traditional way to measure those was by digesting the cell population which had been subjected to, let's say, a nanoparticle uptake experiment, and we get an average value of the amount of nanoparticles in the cells. but that's all what we would get, whilst in the single cell ICP-MS approach, we would be looking at determining the number of nanoparticles incorporated in single cells and get therefore a distribution of values over the cell population with an average and parameters for the width of the distribution. How does single cell ICP-MS work? The typical setup would be an exposure experiment where we would have the cells in a nutrient medium, and those would be exposed to either the nanoparticles or the small molecular drugs, which would be the subject of the test to see to what extent those get taken up by the cells. After the uptake experiment, there would be a separation step which would remove the dissolved or the residual suspended nanoparticles, in the case of nanoparticle materials, from the cells, and those would be washing steps which would be repeated a number of times in order to make sure we end up with a suspension which only contains the cells. And those cells are then sampled with an uptake line to a ICP-MS system, which we see here symbolized by this box. The major component of the ICP-MS system is the plasma, which generates the conditions necessary for dissociation of all the components in the cell into its individual atoms and ions. And those ions are then sampled with an interface which routes those ions into the mass spectrometer. So those ions are then undergoing mass separation, and the target mass is then selected within 1 AMU unit at the detector, and we get a distribution of, we get a time-resolved distribution of signals corresponding to the measurement of the destruction of individual cells in the plasma. So that's those time signals you see here. So if any particle or if any cell does not contain any small molecular drug or particle, then in that case we actually don't see a peak. So that's the case for the this item here. So we have a cell which comes into the plasma, does not have the target element incorporated, and therefore it's just a flat baseline at that point. But however, those cells which do contain the target element, they will be measured as individual transient peaks. And so the intensity of the pulse is then related to the mass of the target element in the cell. So that would be corresponding to the uptake of the small molecular drug when we are doing solute experiments, or to the number of nanoparticles in the cell if you do nanoparticle experiments. And also the number of pulses are of importance because they are related to the number of cells which contain the target element. The baseline is giving an indication of the dissolved background of that target element. Now let's take a step back from a single cell and talk about single particle ICP-MS for a moment. This is to better understand the differences in hardware requirements for the single cell experiments. In nanoparticle measurements, we have a similar requirement to transfer the nanoparticles in solution to the plasma, where they will create an ion plume, which is then detected in the ICP-MS instrument. The result will be a time-resolved signal at the detector, which shows pulses of 400 to 600 microseconds typically of the target element, and those pulses are then related to the frequency of to the frequency of the particles entering the plasma. That means the number of particles in the solution, and the height of those pulses is related to the amount of target analyte in the nanoparticles. The setup for single particle and single cell ICP-MS is different for the reasons that cells behave different than the nanoparticles. And also the software focuses on different aspects of the evaluation. So the distributions for nanoparticles are reported in size. The distribution for cells are reported in mass. So the software takes into account for this for the two different models. We have a single particle software module and we have a single cell software module. And in order to get the cells into the plasma, we need a specific hardware, as the traditional hardware does not permit cells to reach the plasma in a significant quantity, and the uptake system here is displayed for the single cells below with the special aspirin spray chamber, which has a high transfer efficiencies for the cells, and a specific auto sampler which is ensuring the reproducible sampling of the cell suspensions at the low flow rates required for the aspirin spray chamber. Here's a bit more detail on those two different sample introduction systems. So the cyclonic system, if we look at the blue graph below the cyclonic, we see that the nanoparticles are measured as expected with a high transfer efficiency, whilst the actual larger entities, in this case 2.5 micrometer beads, are largely not making its way through the cyclonic spray chamber, and we get hardly any signal for those. On the other hand, the aspirin spray chamber does transmit the nanoparticles, but it also transmits the larger entities, which would be corresponding then to cells, also allows a measurement of cells. So this is the difference between the two introduction systems, typically because of the uptake requirements for the aspirin with its high transfer efficiency. You would not do nanoparticle measurements on that systems, but you can. So in this case, you still would normally switch between the two systems depending. But since we are in this talk really talking about the cells, we are really talking about the aspirin spray chamber in order to ensure an effective transfer of the cells into the plasma. And that is described in the tech note, which is was highlighted here in the slide. So let's go on to the software differences. So the difference, well, let's say the acquisition is also characterized by determining a target element mass with a certain acquisition rate. And those parameters are displayed here in the method parameter section. So those are the acquisition details. We would calibrate typically the signals which are generated in terms of mass by having a separate section here in the software which is dedicated to the calibration, and the real-time signal is then the resulting live feed coming from the ICP-MS in terms of its detected signal. So we have counts here as the unit which is indicative of the magnitude of the signal, and we also see the frequency here as it's accumulated. And this real-time distributions are then converted into results, which means the applied calibration is converting those raw results into amounts of target element in the individual cells which are being measured, and the frequencies transferred to a distribution, and those quantity distributions are then expressed in numbers in terms of average amounts and the corresponding frequencies of the cells in the suspensions. Okay, with that, let's move on to the applications. Having had a review of the requirements in order to do those measurements, we want to look at some of the work has been done on the small molecular drugs uptake in cancer cells. Here we look specifically for cisplatin uptake. Cisplatin is important in cancer because the decreased cellular accumulation is one factor for cisplatinum resistance to treatment. And traditionally, it's the situation which I've explained earlier, that we have a whole population of cells which is examined, but we would like to basically find out more information about the cell population, and we would like to answer those question marks here of which path the uptake experiment is taking. Are we having equal quantities of cisplatin in the exposed cells, or are having a strong or medium wide distribution? And those questions are important to allow answers on the treatment effectiveness which can be expected from the drug. So here we would see a typical uptake experiment. So the cells are exposed, in this case to 13 micromolar cisplatin, and the cells are sampled at various time intervals after exposure. And we see here with those distributions, and that the content of cisplatin in the individual cell, number one, varies. And we also can see that the distribution varies with time. So the platinum content of the cell increases with time, but also the distribution becomes wider. Repetition of those experiments with cisplatin resistant cell lines was able to show that the resistant cell line takes up less cisplatin than the cisplatin sensitive cell line. And an additional test with serum starved cells was aiming to answer the question if the cell cycle which the cell is currently in is responsible for any of the cisplatin resistance observed. And those tests were showing that the cell cycle is not responsible for the observed cisplatin resistance effect. So in this respect, a number of control experiments were basically included, and all of this is published in our app note, which was done by my colleague Chadi Stefan in collaboration with the researchers from the National Institute of Health. And in summary, we are now able to get insight from those type of experiments at the single cell level, which was not possible before. Let's move on to gold nanoparticle uptake in cancer cells. The interest comes as the nanoparticles have attractive benefits for drug delivery to overcome limitations in conventional chemotherapy. The engineered nanoparticles in this respect offer typically advantage over the conventional small molecular drugs in terms of the circulation time in the body and the reduced non-specific cellular uptake. So we can target the affected cancerous tissue more precisely with the nanoparticles, and improve cellular interactions by targeting specific cancer cell areas. The outset for the experiment is, as we had in the previous situation for the small molecular drugs, we would like to get information on the single cell basis instead of just an average for the whole population. And in order to get this information, we again do an uptake experiment. And in this case, the result would show histograms of the mass distribution of gold for the individual cells which gave a signal. And so we see the frequency distribution on the right in the case of A for individual T24 cells. And the interpretation of those type of distributions is related to the number of nanoparticles which have accumulated in individual cells in order to explain such mass distributions. And that's the interpretation which is in B, in slide B, in graph B. And we have the corresponding nanoparticle numbers per cell in this experiment. So we see most cells contain, there's no cells which contain any gold nanoparticles contain 1 nanoparticle, and but there's a significant number which contain more. In this case, up to 5 nanoparticles per cell. So with this type of experiments, the questions on efficiency of drug delivery can be answered because single cell ICP-MS allows the quantification of the number of cells that contain the drug, and also allows a prediction of the treatment efficiency, as we can quantify the number of gold nanoparticles taken up by individual cells. So it is related to the dose per cell. And it also will therefore play an important role in drug discovery, as the single cell ICP-MS technique is then an essential tool for nanoparticle-based cancer therapeutics. This work is again taken just as a very short summary from the much more extensive upload, which is shown here on the right, again downloadable from our website. With that, I want to move on to a third example, which is boron neutron capture therapy. This type of therapy involves boron-based reagents which accumulate in the cancer cells. And the large capture efficiency of boron-10 would then allow the local production of all alpha particles in the cancer cells by the disintegration of the boron-10. And this technique requires therefore selectivity for the boron reagents for the cancer cells. And in addition, it requires knowledge of how much boron is in the cells. A great deal of research focus is going at the moment to understand better the increased, or how to better increase the boron content in the cells. And therefore the need exists to measure the boron reagent quantity and its distribution in single cells. Looking at the instrumental capabilities which are required to support those type of experiments in neutron capture therapy, is needing to take into account the interferences which are needing to be controlled or eliminated. So if you look at boron, or if you look at sulfur, another element which I want to single out as a potentially interesting element for those type of therapies, we would look at interferences from carbon for boron as it's in neighboring mass, or for polyatomic ions in the case of sulfur. And the high abundance sensitivity would ensure of an instrument, ICP-MS instrument, would ensure a low background, okay, in the neighboring mass of the target analyte in order to preclude false positives. And the alternative would be to measure at the mass which is one further mass unit away from the interfering high abundance mass. And this type of questions can be addressed with the appropriate instrumentation. For sulfur, the polyatomic ions would typically be controlled. The interference by those would typically be controlled by shifting the mass of sulfur away from those polyatomics to a higher mass. And it's a different type of approach. And we would basically look at the required functionalities in the ICP-MS for this type of experiment. And this type of discussion I want to do regarding the NexION 2000 and 5000. So 5000 is now coming up to 2 years on the market. We introduced it last year, beginning of last year. And we want to, at this point, introduce also the new instrumental capabilities which can maximize performance for those type of experiments. We have a lot of experience in biomonitoring applications using the NexION product line. Let's have a look where the NexION 5000 can make a difference compared to the NexION 2000. When we talk about biomonitoring applications, we talk about urine, serum, blood analysis typically. We might be talking about single element analysis, toxic panels of elements, nutrient panels, and also speciation of several toxic elements, arsenic, chromium, mercury, or particle analysis in body fluids, like for example for titanium. And what's common there is a situation where we have a matrix of high salt, high organics, high protein samples, which might give rise to certain interferences, which we need to control or eliminate. This is an example where NexION 2000, NexION 5000 differ in terms of its instrumental setup when we want to do measurement of a target element, in this case manganese, which is situated next door in terms of its mass to a high abundance isotope, in this case iron. So manganese in blood would need to be made with an instrumental setup where we control the spillover of the neighboring high mass onto the target mass. Spillover in terms of ICP-MS is characterized by a parameter called the abundance sensitivity. So the instrument would need to have high abundance sensitivity. And that is one aspect. It also would need to control the interferences which can happen at the target mass, in that we talk about reaction chemistry in the cell. So it's the dynamic reaction cell which comes into play here. So there's two aspects. I just want to highlight really the aspect about the abundance sensitivity, which is in the NexION 5000 taken care of by having two analytical quality mass spectrometers in the system with an additive abundance sensitivity of each of those. So we have a very strong separation between the individual AMUs, and we can measure the target element as it's shown here on this graph for NexION 5000, where we see no spillover at all onto the target mass of mass 55. In the NexION 2000, we would have to make sure that we have sufficient resolution, custom resolution on the mass 55 that helps in order to avoid any effects of that nature. In the next 5000, we are not restricted to resort to those changes in the resolution set up from the default, which is 0.7 for those systems. And we see here the ability to measure part per trillion level additions to the native manganese in the samples. So the detailed comparison for the NexION 2000 and NexION 5000, I want to attempt here with a side-by-side comparison of the main technological outline of the main components of the instrument. As we can see here, there's quite a lot the instruments have actually in common. So they have in common the Q0, which is the optical part of the ICP-MS with the function to direct the ions from the cone interface to the downstream MS system. We have in common the cell, which has the job of eliminating interferences. In both systems, it's a quadrupole-based cell with the capability to exert a stability field in the cell to reject ions which are not of interest. And further downstream, a analytical quad, which is measuring the target analyte. NexION 5000, in addition, has a quadrupole in front of the cell, which is restricting the range of ions to within 1 AMU unit of what's then allowed to entering the cell, and therefore able to take part in any reactions which we might want to promote in the cell in order to eliminate interferences. So in the case of the neutron capture technology, as I've mentioned earlier, interferences are interference removal by mass shift is a situation for sulfur, possible application for sulfur. So the differences between the models can there be decisive, let's say, in which interferences to control. If you look here at sulfur by mass shift, main isotope would be 32 of sulfur. If we measure 32 by mass shift with oxygen, we end up at mass 48. And we can see here that titanium is a possible interference, as is calcium. Titanium would be actually reacting away in the cell by the same reaction with oxygen, would shift to a higher mass. Calcium does not. So in this case, the NexION 5000 would have an advantage in terms to take care of the interference by calcium because we can exclude it from entering the cell. However, the main ion of interest is sulfur 33, and if we can measure that directly, we can do that without the interference of calcium, and the titanium, as I mentioned, reacts away, so we actually can measure sulfur 33 interference-free on both instruments. However, if that is a feasible reality in the experimental setup, depends on the quantity of sulfur 33 involved, and so the decision would have to be made based on that information. But with that, I want to move on to the last distinguishing, well, to the second main distinguishing feature for the NexION 5000, and that is the omniring, which is present as part of the cone, triple cone interface. So the cone assembly includes an omniring, which is having the function of providing better transfer efficiency of ions to the downstream MS system compared to its NexION 2000. So we end up with higher sensitivities on the NexION 5000 compared to 2000, and therefore improvements in quantitation. So the background of both models, here you see 2017 was the NexION 2000 introduction, 2020 NexION 5000 introduction. We see the sensitivity increasing by 5-fold, The background is well controlled in both systems, smaller than one, and so the signal to noise goes up on the system, and better detection capabilities on the 5000 compared to the 2000, which might be consideration in addition to the technological considerations outlined earlier for the cancer research applications. And the key benefits for the 5000 would therefore be outstanding interference control, detection capabilities, sub-PPT BECs, and the throughput advantages of the NexION product line in general, with its outstanding long-term stability due to its interface QID design. And that will eliminate a lot of maintenance and therefore increase productivity. So we have a few shared features also of the NexION series, which I want to highlight in the following yet. With that, I want to come to a key slide for this talk, which is the list of, let's say, a wish list of features we would like to see in an ICP-MS in order to make it high performance for single-cell ICP-MS applications. An ICP-MS of this nature would need to have a robust plasma with high power coupling efficiency to deal with high matrix solutions. Also, the interface itself needs to be matrix tolerant in order to avoid drift during our experiments. Sensitive instrument is desirable with low instrumental backgrounds to get best detection limits. We want to remove interferences which might be present on our target analyte. So we want to have a cell which is capable to lower and eliminate those type of interferences. So the background reduction with reactive gases is typically the choice in this situation, as reactive gases are more effective than collision gases to remove interferences. And our target analyte is well known, so these are ideal situations to favor reaction chemistry. So a system which is proven in terms of its performance for reaction capability is desirable. Multi-quad capability is going in that line, is providing maximum accuracy. Some analytes are benefiting especially from this. And then there's the lowering of detection limits, which the NexION 5000 in this case as a multi-quad brings. Fast data acquisition speed is desirable because we're dealing with transient signals which are generated by the plume of ions which are released by single cells when they hit the plasma. And low maintenance and high instrument uptime are also desirable in order to carry out a sequence of experiments without needing to resort to cleaning procedures or replacement procedures during that time span. Furthermore, we want an adequate sample introduction system which is capable to transport our cells into the ICP-MS system, into the plasma, and software which is able to handle the data acquisition and data processing. The NexION 2000, 5000 series are satisfying those criteria optimally. The aspirant single cell autosampler combination is taking care of the aspect of the sample introduction, and the software requirements are taken care of by the single cell app. And so the ability of the NexION product line to provide a complete solution for single cell ICP-MS applications is given. So how can I make these claims, which I made in the previous slides, that the NexION platform is superbly equipped to make SC ICP-MS measurements with high performance? In order to answer that question, we really need to look at the feature which distinguish the NexION platform. I want to do this here quickly in a bullet point type of list. Number one is the universal cell technology, which allows specific control of reaction chemistry. Number two is the MS-MS dilution, unique to the NexION series, to attenuate signal ions which are above range for the mass spectrometer. This is complemented by an all matrix solution, a gas dilution system, which is desirable to have possibly for high matrix suspensions, cell suspensions, which are possibly entering the mass spectrometer. We can dilute those with a gas dilution online prior to the plasma. A triple cone interface is providing a system which is maintenance-free behind the cones, so no maintenance at all in the mass spectrometer system. The industry first 34 megahertz solid-state free-running RF generator is now three or 4 years on the market. It's been completely redesigned RF generator, which is specifically geared for reliable performance in challenging matrix. The LumiCoil is again a novel RF load coil with on the market now for 4 years to exert a lower demand for the user interventions and for maintenance, and the smart intro systems, color-coded introduction systems for the sample intro system allow in a multi-user environment to have the possibility to set up within minutes the ICP-MS system to the application which is desired, in this case the single cell ICP-MS application. I want to especially highlight the feature of the universal cell technology. With that we can measure in reaction mode, collision mode, or standard mode. I want to highlight dynamic bandpass tuning, which the universal cell features, and axial field technology, providing control over the reactions with the dynamic bandpass, which is able to eject unwanted ions from the cell, and with the axial field, which allows controlling the speed of the ions in the cell. So we have a tool in this case which is providing a lot of flexibility on how to set up an experiment for single ICP-MS experiment analysis. Collision mode is characterized by generally less specific interactions and loss of analyte signal, especially at lower masses, very significant. And so if we need to remove interferences, we have to select either of those two. If we don't have interferences, we can use standard mode. The case is that in single cell ICP-MS, we typically know our target analyte very well. We have only one typically, and the interferences are also well characterized, so it's possible to select a reaction gas and therefore benefit from the lower detection limits, lower BECs, the higher efficiency which reaction mode offers. Let's look a little bit more detail of this. So this is an example where we remove an interference with reaction mode. We see here the high intensities of a matrix, of a polyatomic interference from the matrix. being reduced to very, very low levels very fast if we bleed a small amount of reaction gas into the cell. And we can then distinguish the analyte on top of the very low background if the cell gas level is sufficiently high. So very efficient removal of the interference. This is different for collision mode. In collision mode, we typically have a much more gradual removal of the interference and therefore more loss of analyte signal, and the efficiency is more gradual. It's not as clear cut as in reaction mode, and the resulting performance not as good. So the second important point is the interface. What distinguishes the NexION series is a triple cone interface. A triple cone interface provides a more tightly defined ion beam. We see that here in the second panel. So we have here the sampler cone, skimmer cone, and hyperskimmer cone. And in that area, sampler, skimmer, and hyperskimmer cone, which provide a narrow ion beam in the order of 2 to 3 millimeters behind the cones. That allows maintenance-free QID operation, maintenance-free optical system. That is not the case for the typical competition systems on the market, which consist of 2 cones, which have a much higher beam divergence for the unionized and ionized material, and the resulting ion optical system therefore is contaminated more heavily and needs to be cleaned with. Final slide on the sum up. The NexION user-friendly aspect is about maintenance. There's a lot of maintenance tasks which do not need to be done on a NexION, which need to be done on a competitor instrument. This slide is giving an overview of those. Key is there's no maintenance behind the cones, so you don't have to break vacuum. There's a lot of individual factors involved in that maintenance performance for the NexION, and that relates to the cones, which do not be cleaned as frequently. The extraction lens, which is absent in the NexION, does not to be cleaned for that reason. The ion lens, which is absent in the NexION. We don't have a traditional ion lens. We have a QID instead, which is maintenance-free. The pump oil is of a high quality. Oops, should take that to my pointer. The high quality oil is giving a replacement time up to 60 months, so up to 5 years. Collision reaction cell does not need to be maintained or changed in the NexION in the product line. The detector has a longer lifetime, and the torch shield is non-existent. We don't use one, we don't need one, and for also eliminated that consumable, as well as the RF coil, which is for the lifetime of the instrument, which is not the case in competitor instruments. With that, I want to just move on to the whole area, how that is not only important in terms of the frequency of cleaning, but also important in terms of the drift. So in a typical NexION instrument, the hole for the interface is much smaller compared to the NexION, and that means that when we coat that cone with high matrix, the relative diameter will change more in a competitor instrument compared to the NexION. That means the drift will be less on an NexION, and for that reason we have the best drift spec on the market for the NexION, important parameter for the single cell ICP-MS experiments, which need to be done typically in sequence for a number of cell lines, for a number of different experimental setups, and we want to avoid changes in our signal intensity during that experiment. This view just gives us a visual demonstration of how significant that change in whole area actually is. Okay, with that I want to sum up. I want to basically highlight that we talked about single cell ICP-MS creating new options for quantitating elemental distributions at the level of the single cell, that such experiments require robust hardware and integrated software to facilitate routine measurements. The ability to look at solute uptake and nanoparticle uptake in single cells benefits a variety of medical settings, and with the new product line addition of the NexION 5000, we have now new instrumental features available which maximize performance for single ICP-MS for promising research, elements in cancer research. And I singled out in this talk especially neutron capture technology application, neutron capture therapy applications involving boron or sulfur. And with that, I want to thank you for your attention, and I shall join you now live for a Q&A system, and hope you enjoyed the talk. Thank you very much for your attention.
Okay, thank you very much, Helmut. That was fantastic. I know we are at the hour, but let's, I think, a few people are still sticking around. Well, actually, most people looks like. So I have time, I think, to help answer the questions. Helmut, you're still here, right?
Yes, I'm here. Yep.
Excellent, fantastic.
Hello, everybody.
So just reminder, please put questions in the questions panes and the panels, and we'll answer them. So starting with the first question, Helmut, how is the acquisition time determined? I'm guessing that's during single particle or single cell analysis.
The acquisition time is determined by how many events you need to measure during that specific measurement. So this is relatively similar to single particle ICP-MS measurements. You have a certain number of entities, particles, cells in the suspension, and you typically want to take the quantity of cells which enter the mass spectrometer to be in a range of around 50,000 cells per milliliter, where you have basically sufficient signal, but not too much signal. You don't overlap those individual discrete events. So as we have a time resolved signal, we need to make sure that those acquisitions are done with minimum interference. So we need to basically set up an experiment in that way, and you would collect sufficient time, in order to have a distribution which is well characterized. So the width of the distribution plays a bit of a role. If you have a very well-defined distribution of material in a cell, then you can probably live with a shorter acquisition time for the experiment. But generally, the increased time allows you to count more cells, and therefore better define your distribution.
You have better counting statistics, right? It all comes down to counting statistics ultimately when you're counting things, right?
Yeah, it's counting statistics also for the event of the that we are reaching sort of levels of the target analyte which are let's say not as well distinguishable from the background. In that case, yes, counting more helps. Counting for longer helps. Generally speaking, also each individual cell, even if they have large counts, with large quantities of material, will still be resulting in a better definition of the distribution if we count for longer, because we just collect more events during that time. So That would be the considerations to take into account. So you're right, Aaron.
Yeah, I mean that the basics I always try and explain is
absolutely, yeah.
Yeah, and then yeah, obviously there's other factors like signal to background and stuff like that too.
Yeah, absolutely, yeah.
So for
I mean that's right.
Sorry,
So since we
I just
with
kind
the
of
mass
moved
spectrometer.
to the next question. We have a few here to
Okay.
get.
Yep.
For single cell analysis, what is the expiration date of a commercial standard?
Well, we would actually calibrate our experiment frequently also with nanoparticle solutions too. So those expirations are not as time critical. There are expiration dates, which I can't tell off the top of my head, but I think they're in the range of 2 years or so for those standards. So it's not very time critical.
It depends on the metal too, and depends on you know the suspension, the size, the capping agent, the buffer. This there's a variety of kind of data. Most nanoparticles are like only a few months, like 6 months to 12 months. For single cells, it's tough, right? Cell, they're living thing, right? So, so that you want to use them as fast as possible. If you've exposed them to metal or nanoparticles.
Absolutely.
Right? Yeah, it's I've seen a lot of experiments go bad letting it wait too long. So
absolutely, yeah. So yeah, and so in terms of the lifetime of the standards, I mean, if you have characterized your system, well, let's say with one type of setup, there's nanoparticles are one way. There's also the way of coated beads with rare earth metals, which can be used for this. And once you have defined your system and your transfer efficiency sufficiently, it's frequently also a matter of using dissolved standards, which are even easier to use to complete the calibration.
Yeah, and I've even done single cell calibration, like transport efficiency calibration, using intrinsic, metals that are intrinsic to the cell, such as zinc or iron, and zinc being one of the main ones. And then I use a cytometer. We've used, we've grown the cells, used a cytometer, counted the cells, diluted it down to appropriate cell count, and then use that cytometer data to kind of calibrate as well, using the intrinsic zinc content, for example, has been one. But yeah, so there's different ways of going about it. As Helmut says, you have to define that as part of your experiment. Next, is it possible to monitor more than one element in a transient signal that lasts less than a millisecond?
Well, we do have a dual analyte capability for monitoring at the most 2 isotopes. More is not 2 elements. More is not possible. Typically, as I said, experiments are done for 1.
Yeah. As soon as you hop off that mass, though, you lose some data quality on that event or the events that you're moving from. But we have been looking at it more so for understanding like metal alloy particles and stuff like that, or shell core particles.
Yeah, in single cell ICP-MS analysis, I have not come across the need.
Not in single cell, no. The problem with single cell is you flow. It's not as simple as single particle, really, right? Because you have to make sure that those cells are being introduced into the mass spectrometer intact, and so you don't have very the freedom as much as you do with single particle just to change things up quickly. And then just a bit of a comment with the question. So thanks for the nice presentation. I have done single cell ICP-MS of platinum inside cells cultured with platinum-based drugs using the NexION 2000 and single cell application. The intracellular contents of platinum measured were clearly lower than the ones obtained by the traditional method with acid digestion of cells, although the cells were well preserved before single cell analysis. Also, various washing steps of cells were done prior to analysis. Is there any explanation for the difference between the two methods? I'm assuming the bulk analysis and the single cell analysis.
Yeah, I wouldn't be able to give you a comment based on experience for this. I mean, generally speaking, with the platinum being very high mass element, it suffers less from sort of space charge effects or those nature which can skew signals, because if you don't use an internal standard, if the signal intensity gets skewed by factors which are, let's say, not correctly taken into account in a single cell experiment, then it could lead to deviations. But for platinum, I wouldn't expect that to be dramatically the case. So we would probably have to look a bit closer. Yeah.
We'd have to look at all the variables, right? How's the cell counting done? When you're rinsing the cell, sounds like you're rinsing, then taking it as aliquot, digesting and looking at total, and then taking an aliquot and looking at single cell. Is there any time difference? Was there anything happening there? And then, when you're rinsing, did you look at all the rinsates and see if there was any platinum in the rinsate? So sometimes when we do that, we rinse. We take the rinse solution from it and make sure that there's no target element, in this case platinum. Make sure there's no target element in the rinse to prove that we're rinsing it properly, and that we're only looking at the metal, within the cell rather than around the cells. There's a few variables, and if you're doing, obviously cytometry on it somehow, how's that, what's the error there, etc. Okay, next question. Enjoyed your talk. It's my understanding correct that for single cell measurements, we need a separate software module?
Well, I would say yes, because the single particle software, which is the alternative, is geared for sizes, reporting the results in size distributions. And that is a layer of data treatment which is not relevant for the single cell. At PerkinElmer, we have, for that reason, two modules, because the data treatment is similar, but it's sufficiently different to benefit from a reporting which is specific for single cell.
Exactly. Yeah. Yep. So I guess the simple answer is yes. You need a separate software module to really take advantage of the system, I believe.
Yeah.
There's other factors as well, because single cell ICP-MS, the automation really helps in accurate and precise delivery of the cell suspension. And so another question, in single cell analysis, if there are high dissolved ions as well as particles present in the cells, is there a way to differentiate between the two?
If the background is too high, it will be difficult. The higher the background, the more you drown out your signal from the particles. So the detection limit becomes worse, and you don't see down to the, let's say, quantity levels individual cells, which you would be able to do in the absence of the dissolved background. So you would have to decide for individual experiment if your background level is too high or not to see the cell. Depends on how much you have in your cells of the target analyte. So what's the signal difference between the baseline and the individual discrete events?
If there is If there is a good difference though, right, you can differentiate, you can,
exactly.
quantify to an extent, what's in the dissolved content versus what's in the cell, or, within the event, right, created by the cell. So you can, but you're right, there's the greater the background, it always compromises detection, right?
Exactly.
There's always some compromise, but somewhat from, I'm not, cell expert, but the nice thing about cells is you can do those rinsings, right? So you can rinse the cells as long as you keep them safe and intact. You can rinse them to an extent to get rid of some of that metal that could be dissolved around it, which is nice.
Yeah, absolutely.
So, nanoparticles we just dilute. Cells you can wash, kind of, right? So, okay, then next question. One question about the raw data, the data that's, I'm assuming there's no threshold applied. Can we get it easily in the software / instrument data?
Yeah, the data is recorded for each measurement also as a simple time versus intensity file.
Correct.
So those are basically what they will call maybe metadata. They are used, they are accessible, but normally you would not see them. But you can access them if you like to process your data in a different way externally. That's possible.
Yep. And we have a few customers doing that. So they take the raw data output and they do their own thing with data processing, which is fine. Yeah, it works. Yep. And then I think a comment here is, well, with the shelf life of cells and the measurement possibilities, also fixing the cells could be helpful sometimes. Okay, yeah. That's a good point. Yes, I do know researchers doing that, because, as you mentioned here, cells do have a shelf life. Many of them do, and measurement possibilities, you can fix the cells, so stop them from rupturing, not being viable anymore. Good point. That's a good point. And another comment here, not a question, but still, dual analyte would be useful to make the ratios of elements in the cells, if you would like to have analyzed element and the reference one. So if possible, could be. I mean, that's a good point. Yeah, especially I could see that with single cells. You could ratio things, like intrinsic to non-intrinsic metals. Yeah, that could be, sure, right?
Yep.
Yeah. And you can identify certain things that could be tagged things. So yeah, I think that's a good point. Yeah. All right, that's it, Helmut. We've a little bit over our time, but it was a really good, really educational talk. So thank you. And for the recording of this on demand and other webinars, you can visit atomicwebinars.com. I've set up this website to archive a lot of this. And I just want, on behalf of Park and Elmer, I'd like to thank you, Helmut, and thank everyone for attending. And have yourselves a great rest of your evening, day, week, and take care.
Okay, thanks also from my side, and goodbye to everybody.
Take care, Helmut. Bye.
Bye.